So in one of my later videos, I'm going to be talking a bit about DNA, but I'll introduce some of that stuff right now. So DNA is a double helix, and it runs from 5' to 3'. So I have a 5' that goes to 3', and a 3', which makes it anti-parallel, which goes to 5'. Okay, so this double helix actually has a lot of information in it. It's your genetic information which determines you from one person to another. The process of remaking this DNA is called replication. Replication is when you duplicate your DNA. So what happens is you have two strands of DNA. The 5' to 3' and the 3' to 5'. And what happens is when DNA is replicating, an enzyme known as DNA polymerase can only read off of the DNA strand that is exposed from 3' to 5'. This is my bottom strand. Okay? Now what happens is DNA polymerase binds here and it begins to make your DNA sequence. This is known as your leading strand. And this replicates continuously. This strand is your lagging strand. What happens is as this DNA helix is opening, since DNA polymerase can only replicate from 3' to 5', it has to wait for enough DNA to open up so it can bind at the 3' end and then continue to make your DNA this way. Now each time it does that, for the lagging strand, you get something that we call Okazaki fragments. Say it with me. Okazaki is how you say that name. And it's named after the Japanese scientist who actually founded these. Now, each of these strands that are in green, they're actually going to make their own DNA strand, to make two DNA strands. And each of these DNA strands will go into a separating cell. Later on, I will get into something known as mitosis, which is cell division, and it'll show you the purpose of why the cell needs to replicate its DNA. The other thing that I'd like to mention is before DNA polymerase can actually bond to these strands, you need something known as an RNA primer. And what this RNA primer is, it's a part of RNA that gets inserted by a different enzyme known as RNA polymerase. Because what DNA polymerase can't do is it can't take different nucleotides from random and bring them and connect them to the DNA. Now, RNA polymerase is able to do that, in which case you need the RNA primer to get inserted here before DNA polymerase can start replicating. Subscribe to me so you guys can see some cool videos doing science and experiments and maybe learning something new. Thanks!